Welcome to this session. Sorry for the delay. So this session is called Beyond the Silos, Uniting for Fair Genome Metadata. This is presenting the preliminary outputs from the FAIRification of Genomic Annotations working group. So first off, we want to acknowledge and celebrate the first Australians on whose traditional lands we meet, and we pay our respect to their elders past and present. This is just to remind you of the 6 guiding principles at the heart of the RDA community: openness, community-driven, consensus, non-profit and technology-neutral, harmonization and inclusivity. And all RDA members are expected to adhere to the RDA Code of Conduct. Okay, so I'll start this session with a brief overview of the FAIRification of Genomic Annotations Working Group, or short FGA-WG. My name is Sveinung Gundersen. I'm working at the University of Oslo in the ELIXIR Norway project. And I'm a co-chair of the working group. So first of all, introduction to the domain. So this is about, since it's an open session, domain cross domains, then I'll just present what is the data types that we're talking about here, and for those who are not very much into genomics. So first of all, you can look at a genome, which is the totality of DNA in an individual, as a sequence of letters, basically one of 4 nucleotides, A, T, C, and G, and this is, as many would know, the sort of data content of the DNA itself. On the other hand, in order to make sense of this sequence, you need some other information, and in order to do that, you need to annotate the DNA sequence with the data elements that can come from different experiments or other algorithmical sources. So the main typical example and sort of the most important of these annotations is the positions of genes. But there are a number of other data sets that can be positioned along a genome sequence like this. So a genome annotation, or a genomic track, which is also called, is then a series of related data elements positioned along a reference genome sequence like this. So the reason this data type is important came historically from a visualization type of visualization software called genome browsers, where you can visualize these different genome annotations or tracks as a horizontal line, and then with the DNA sequence as the coordinate system. And then you can look at different regions and figure out what happens, and compare different data sets and so on. There's also another of analysis tools that was created for this purpose, not just for visual analysis, but also statistical analysis, or other types of analysis used to make sense of this type of data. So one of these, just to tell me a bit about my background here, is a software system called the Genomic Hyperbrowser, which we published in 2010, which is a rigorous approach to statistical analysis of annotation data. And this is also then my PhD thesis from some years later, looking at representation and integrated analysis of heterogeneous genomic datasets. So you can say that harmonization of data and analysis is a recurring topic in my career. And then in the late years, I started to focus on the issue of harmonizing metadata. So just to tell a bit more about the background for this working group, it's been a longer process. As I mentioned, we started with this analysis software and started to look into how to import data sets into this system. In order to do that, we created a prototype where we started to harmonize some metadata fields. But we found out that this was too difficult project for us to do on our own, so we started collaborating in the context of ELIXIR. So ELIXIR is a European infrastructure or collaboration across the European countries. And in that context, we started an implementation study with ELIXIR Norway, ELIXIR Spain, and the EMBL EBI, which is providing many of the most important repositories for the life science community. And this ended up in the publication 2021, some years ago, with the proof concept implementations. Then the question arose, what to do now? And now we have this concept. We have developed some recommendations for verifying metadata for this type of genomic annotations. But in order to move forward and get this adopted, we need some way to do that, to get into a broader community. And then we approached the RDA and were selected as a pilot for the RDA Tiger project. And then through that, we managed to get enough interest globally to start a working group that has been running for a bit more than a year. So this is FAIRification Genomic Annotations Working Group. There's also other projects that's leading into this from the other co-chairs. So one example is the Genomic Computing project, which was ran with European money in Italy, mainly Milano. So this ran in parallel to the FAIRtrack project, and in many ways did quite similar things, although moved more into production than the proof of concepts that we created in that project. Another project that is providing some background here which is now being developed currently, called BedBase, which is about extracting metadata from more uncharacterized sources in repositories using AI methodology and providing search capabilities through a number of what's called bed files, which is a specific file format for this type of data, most of which is very difficult to find and make use of with traditional methods. A third project that's also led by one of the co-chairs, Adam Wright, is called FAIR Bioheaders, which looked at adding some metadata to one important file type in genomics called FASTA files, which are the ones that used to denote DNA sequences. So I also forgot to say that the Genomic Computing project is represented in the working group by Anna Bernasconi, and Nathan Sheffield is leading the BedBase project. So all of these are co-chairs for the working group. Okay. So to say some few words about the scope of the working group, there has been a number of major consortia that have produced functional genomic data over the last 20 years. Here are some examples with the timelines, and these have been huge projects. Many of these, for instance, the ENCODE project, in total, billions of dollars have been poured into this project. They produce raw data that's been used by lots of researchers. The problem is that every one of these typically end up with a data portal that is specific to their data type with metadata that is largely incompatible across the projects. And then in addition to this, there's thousands of smaller scale datasets, many of whom have little metadata available. So this is, the last slide was more focusing on the biomedical side. In addition, there's also a large push in biodiversity projects, where millions of new genomes are being sequenced across the world, many of which is under the umbrella of the Earth Biogenome Project. And in that context, there's also a lack of proper metadata or coordinated metadata, and several attempts to get in that direction, which we're collaborating with. So just to precisely specify the aim of the working group, the question to be answered is whether we can harmonize and verify genomic annotation metadata across all of these projects and beyond, and whether we can build a simple community-oriented infrastructure for discovery and reuse of genomic annotations. And this is then the basis of the working group. So just a few more words about the research fields here. So if you look at this broadly, there you can split the life science fields of relevance here into two broad groups. One is the functional genomics or epigenomics area, which is focusing on biomedical research. So mainly humans, also mouse and other model organisms. And what is a important feature of how this group of researchers work with annotation data is that you typically have a reference coordinate system or one line that you compare these tracks or annotations using against each other. I mean, you compare them using the same coordinate system. And then the creation of the assembly and the annotation, the main annotation, the genes for the model, that is typically centralized through some reference genome consortia. So in contrast, from the biodiversity side, the focus is on sequencing a number of genomes and not so much different annotation on one genome. And then look at comparative genomics across the tree of life. So there's often many different coordinate systems used, and these species are sequenced and assembled in different projects using often both centralized but also in many cases distributed pipelines for doing the gene annotation and so on. So even though in principle it's the same problem domain and same type of data, the reality of how research is done is quite different, and the challenges for gathering metadata also is quite different. So we are aiming to provide solutions across both of these large groups. In the working group itself, we have mainly focused on functional genomics and the biomedical side due to the fact that all the co-chairs are coming from that side, working most on this field, while we have then provided some work in the context of ELIXIR in the biohackathon events organized by ELIXIR to reach out biodiversity projects, including the European Reference Genome Atlas, and locally here, the Australian Tree of Life projects also included in this collaboration. Yeah, so one place we started in the working group is trying to figure out some naming and what to actually call this type of data, because there's a Scandinavian proverb saying that the dear child has many names, and there's a number of different names for this in literature and not really consensus. In some cases, talking about genome annotation, other times the word track is used, or data set, or genomic data set, or bed-like data, or a number of different things. So we came up with a term which we call genomic annotations, which is slightly different from a common term called genome annotation mentioned, which is mostly for genes and similar annotations in on biodiversity side, or for the initial annotation. So we're sort of making that term slightly more broad and talk about the genomic annotations, basically meaning any type of data that can be visualized in a one-dimensional infrastructure, one-dimensional coordinate system. So briefly going over the three main use cases for the working group. The first use case that we're focused most on is the discovery of datasets for tools, for use by tools and by AI ML methodology. And the idea is through harmonizing the metadata, not focusing on the data itself. So the data is out of pharmacy data is out of scope, even though there's plenty of things to do there also. We are focusing on the metadata that point to the data files so that you can discover it, and finding the most common fields and metadata records usable for discovery. The other use case is then focused on biodiversity, where genome annotations, that is sort of the initial characterization of a sequenced genome, in the current infrastructure, metadata that relates to that process. So there's this quite complicated process where you run some number of pipelines using data, inference data from other genomes to try to identify genes and other important regions. So, but the metadata for this is largely lost in many cases, or it's not. There's no good process of storing and sharing the metadata. That is being improved in the repositories, but it's still not there. But in any case, there is a need for a community standard or solutions for how to manage the metadata in this case. And this is also identified from the Earth Biogenome Project, from a report that came out 2 years ago. And we're working also together with that project. The last use case that we set out to work with is to enhance something called the Track Hub infrastructure. This is an existing solution that's set up to provide data sources to genome browsers. Unfortunately, we have not really progressed much in this use case due to some key people switching positions. So hopefully we'll be able to get back on track on this. This is then following up on the earlier collaboration with what's called the Track Hub registry at MBLBI. Okay, so that was the introduction for the working group, and the rest of the sessions I will present some preliminary outcomes from the working group. So in order to structure this part, I'll just take you briefly through an outline of the infrastructure that we are in the process of designing for how to set up this community-oriented collaboration and metadata sharing. So first of all, central here is the need for a minimal metadata schema, which is the main output that we will present afterwards. And then when we have that schema, we will try to set up or provide some guidelines for how to enable scalable and maintainable transformation pipelines from the different sources where the metadata currently exists. And here the challenge of scalability and maintainability is often making this difficult, how to make sure that such pipelines can run for a long time. Then there is the need of a strategy for providing persistent identifiers. And then lastly, there's the need for a strategy for how to provide persistent and public deposits of this harmonized metadata. So the idea is that once people start harmonizing, once this metadata is out there available for use, then different third-party services can harvest the metadata, provide search APIs. And the last thing that we are looking at is whether we can provide some uniform search API so that we don't end up with the same problem in the end, that different services provide different ways of discovering data. And once all of this is set up and there is some use of this, then we envision that the researchers start making use of this and then can provide extract data sources, refer to where the data source came from, and probably in the new publications they can then take the outputs and harmonize them, describe them into a system so that you get this research data reuse loop. But that is sort of a bit into the future now. Okay, so first, one of the co-chairs, Anna Bernasconi, will introduce the metadata model, which has just been released in the initial version. And just pointing back to this overview, this is then the central part of this whole infrastructure here. Anna will not be here in person, but she's provide the recording.
Hi everyone, welcome. It is my great pleasure to present you the work on the metadata model for verification of genomic annotations that we did in the context of our verification of genomic annotation working group. So first, we were aiming to develop recommendations for a minimal fair metadata schema. The goal of the working group was to find a way to propose a minimal fair metadata schema for genomic annotations that build on existing FAIRtracks metadata schema and models from other projects such as IHAC, Gecko, FANG projects, and could be evolved to support FGA working group use cases. So we started with making general recommendations for supporting the fairness of metadata on genomic annotations. Our activities comprised a comprehensive review of existing metadata models and use cases and harmonization of current metadata models and extensions as needed by use cases. So we started with this review of existing models. You see here a bunch of them. Some of these already try to integrate across projects. Others are more specific for single projects. Specifically, we looked into two of these models. The one called Gecko, Genomic Conceptual Model from the Data-Driven Genomic Computing ERC Advanced Grant project that includes about 8 entities collected around the idea of item or sample, which is our genomic annotation file. Whereas instead, FAIRtracks minimal metadata model has only 5 entities: the experiment, the study, the biosample, the track within track collections. The two models have many entities and concepts in common, and we tried to extend, build up on this initial basis. First of all, we reviewed some use cases. We started collecting all the experience that the members of the working group had could bring to the community, and we collected a bunch of these, which I am proposing to you. We would like our method model to support all these processes, to search across repositories for relevant annotations, to find both signals and kind of annotations from the same API. We wanted to find, for example, epigenomic signals and also population variations from the same API. We wanted our services to be able to download large data sets with pre-standardized metadata. The metadata model should support an API specification for biomedical data repository. We would like to share uniform metadata for biodiversity genome annotations. We would like to determine a coordinate system compatibility for downloaded bed files. We would like to allow custom schemas for non-human metadata, to pose a focus on provenance of genomic datasets for publications, for example, to access to standardized variant specification, to find dataset that is able to answer research queries in general, and make genome annotation generally findable. So we started putting the focus on the central entity, the central unit of information, which is a genomic annotation. And we analyzed 3 very important dimensions, which are its format, its interpretation, and its geometry. For each of these dimensions, we were able to collect a bunch of different references and data sources that are already offering some kind of taxonomy of values. This is a case of the EDEM ontology for the format and a survey made by Gundersen and others that already mapped formats into their representation. For the interpretation, we relied heavily on the ENCODE proposal, which is already collecting very many type of signals. For the geometry, for example, we relied initially on a proposal, again from the review of Gundersen and others, that already cataloged all the possible shapes, geometry that a genomic annotation may have. And then we enriched this information with the GA-WG DERS and other data access standards, and also a further selection of ontologies, including those chosen within the GA-WG, the ARDC kit, and the suggestion made in the FAIR cookbook. One mention has to be made about the sample and donor entities, which were not the very central focus of our working group, but were very useful anyways to describe the biological material that is being analyzed and processed to provide genomic annotations. For the sample and donor entities, we relied heavily, we based our proposal on the ENCODE one, as in our opinion, this already provides a very complete and precise definition. So here you see our proposal for the entities with a list of attributes, each of which has a description and examples. Then we complete our proposal with all the entities of the FAIR metadata schema that we propose. Here you see the full list. I'm going to present some of these. So the deposition presents a top-level information about an entire metadata submission. The file collection is, as the name says, a collection of files. These could be typically genomic annotation files, but also other types, such as raw data files, such as FASTQ. A study is a scientific study, which is a unit of research for us within which experiments and/or analysis have been carried out. A term is just a helper entity to represent an ontology term as a data value. Then we have input sources, which are general objects representing the source of data files, samples, or other entities. We have contacts, which contain contact information for persons or organizations involved in the experiment. The experiment indeed represents a sequencing experiment that has been carried out within a study. And for this, we relied heavily on the GA-WG experiments metadata model. We aligned to that. Analysis instead represent computational processing applied to data from a sequencing experiment or from another analysis. And this can be described at the level of individual analysis steps in a workflow pipeline, or more generally for the workflow pipeline as a whole. We have then files, which are a general entity. This is taken from the GA-WG DERS object model. So we are aligning to that. And this also applies to the following three entities: checksums, access methods, and access URLs. These are all inherited from the objects from the GA-WG DERS data model. We have then quality assessment, which represent the results of a quality assessment carried out on a data file resulting from an experiment or analysis. And then we have our central entity, the genomic annotation file, which is a specific kind of file representing indeed a genomic annotation. Finally, we have the genome assembly, which provides information about the exact genome assembly that is used to generate the annotation file, defining the genome coordinate system for the sequence features. We note that this entity is prepared in accordance with the GA-WG ref-get sequence collection standard to which we aligned. Here you see the proposal of our schema in a UML format. We have many different versions, but this has been prepared and simplified as a final proposal. You see all the entities that I just presented, including the deposit, the file collection that includes file. One type of file is the genomic annotation file that includes the genomic assembly and the track geometry. The input source takes value from samples that take value from donors. We have the experiment that includes studies and analysis. As a version 2, we are currently looking into open data linking. So we are going to use LinkML, the linked open data modeling language, for maximizing interoperability. Here, finally, you find some useful links about review of existing models, our use cases, the UML diagram versions, and the LinkML implementation, which is ongoing. And thank you very much for your attention. I'm grateful for this opportunity of presenting, and I salute you.
Sorry, I need to just go quickly through the slides. Okay, so I will say a few words about the next output, which, and this is also relatively preliminary for the idea is to also base this on experience. Well, we haven't really gotten to the point where we have implemented much of the transformations. So just to remind you, this guideline is about, this output is about guidelines for enabling scalable and maintainable metadata transformation pipelines. As I mentioned earlier in the presentation, there's a number of different sources for the metadata in question. And the process of transforming metadata at scale from this is something that we have investigated in earlier projects, and there's a number of recurrent steps. So one of the guidelines or outputs would be very much about looking into how to generalize and reuse existing solutions, not to sort of implement everything from scratch in every case. And there's also needs for solutions for sort of scaling this up when for larger file sizes, and how to do that in a structured manner, where to run the pipelines, etc. Automation is something that could be very much looked into. But most importantly, I think, is some ideas about how to maintain the different aspects of a pipeline, when there's lots of fluidity. So, for instance, the source metadata might change, the schema might change, the ontology might change or will change, and how you do the mapping also itself. So version controlling of all these aspects. One solution that we have been developing in the context of ELIXIR in Norway is something called OmniPy, which there also will be poster of here in this conference. So this is a Python library for helping out with a number of these tasks that are often needed for transformation, both of data and metadata in this case. And I will not go much into detail, but this is a framework that we are looking into. In terms of the actual guidelines, I'm sorry that I have not so much to present to you here, because the work really starts now to gather the experience to generate these guidelines. The next output will be a strategy for identifiers. So this is based on some slides from Nathan Sheffield, the co-chair. Unfortunately, he was not able to present, so I will try to go through this. He's been mainly a driver of some of these concepts. So I think one of the things that we have been discussing and looking most into is what we call the difference between authority-based and content-derived identifiers. And I think in the RDA and other places, identifiers are very much the type of identifiers that's very much discussed, even though there are different, of course, groups, whether they are persistent and global, unique, and so on. But in most cases, they are based on authority, meaning that there is some community agreement, formal or non-formal, to follow some authority that provides these identifiers. And the persistency would then be the responsibility of the authority. So what we have been discussing and also following up on developments in the Global Alliance for Genomics and Health, which is a standards creating organization working on genomic data, is the concept of content-derived identifiers, which is distributed in a completely or fundamentally different way than authority-based identifiers. So instead of agreeing for the community to agree on a specific authority, the community agrees on an algorithm or a solution for calculating a digest from the actual data. And the important question then is to identify which qualities of the data file, and this is typically this is specific to specific data types. What are the parts of the data file that inherently represents or identifies the data, so that if you want to find the same data elsewhere, which parts can change, which part has to be the same. So one example of this, which I'll come back to, is a new standard from FGA-WG called the RefGet sequence collection. I'll come back to the next slide. We're going more into detail how one could calculate such content-derived identifiers. As an output, we will probably recommend the generation of content-derived identifiers for annotations or for data files, but probably will not be able to develop or more than possibly a simple example of that. So that will be for follow-up. So this, just to provide you with an idea how a content-derived identifier might look. So this is then from the RefGet sequence collection, which is a standard that was released this year. And basically, this follows upon an earlier version of the standard called just RefGet sequence, where each, and this is for providing identifiers for reference genome or coordinate system or different aspects of the reference genome. So basically, this is a sort of two-step process. So first, the sequences themselves, DNA sequences, they are using an algorithm to create a digest that is uniquely representing one particular sequence. And then, as a second level, we can take these digests and other information as the names of the sequences and other attributes that are of relevance to. Then we combine it together in a more complex algorithm and create one top-level digest that represents the whole collection of sequences. So the sequence collection is due to the fact that we have not just one sequence in, for instance, human DNA, but a number of chromosomes and other alternative sequences and such provided. Importantly, it's also support for coordinate systems and other things. These are a bit domain specific. The main idea here is the idea that you can take a data file generated and with different variants in terms of how it's actually represented, extract it and calculate the same identifier without having to look at or contact an authority. The last output I will go a bit into is a strategy for persistent and public deposition of harmonized metadata. There's very much following the FAIR principle A2, that metadata should be accessible even when data is no longer available. What we would like to propose here is that all metadata that is harmonized according to this model, that the harmonized metadata is published in a general repository so that you get the DOI and it can version control it and pinpoint exactly where it is, and even though the data might be available, the metadata at least will be there. Of course, we could also push for storing the data in a central way like this, but the data might be much larger, and so that's a scalability issue. So at least publish the metadata. Some guidelines: such metadata deposits should support versioned and version-agnostic identifiers. So both of these are very useful in order to, depending on whether you want to point to a particular deposit or at a certain time, or this are the whole stack of deposits over time. And another recommendation is that the metadata deposit should allow pre-registration of DOIs, so that you can include self-referencing DOI in the deposit itself. Right. Sorry. This is actually following on recommendations that we were presented in the FAIRtax project, and on top of that, we are looking into adding onto this. So one thing is to look at the metadata model for a deposit, and I'm sure that this is one thing that we are very interested in getting more information from other RDA working groups. For instance, the metadata model that's inherent in DOI might be very useful. So we want to have a simple model that just describes the deposit, and this is not something that we want to make ourselves, because that at least exists. And we're also looking into solutions for granular file-specific DOIs. We're following a bit the data granularity working group in RDA, and then also investigating the versioning of DOIs in more general terms, as I mentioned a bit before. We also looked into 3 possible solutions for how to create a registry of metadata, because once people deposit metadata, you'd need to have sort of registry in order to locate them. And for this particular domain-specific registry. We want to make this simple. We looked at 3 different options: implement a simple database, possibly have just a single deposit in, for instance, Zenodo, let it act as a registry. What we have come up with as a recommendation is to, and of course this is a bit technology based, but the concepts could be the same in other implementations. So in particular, to have a GitHub repository that could provide the simple metadata about the deposit. So every time somebody harmonized some metadata, want to have published it somewhere, then they take the DOI, take the associated small metadata referring about that deposit, and provides a pull request to the GitHub repository. And then you have a community that sort of approves this and make sure that this follows whatever standards are there. In addition, then there is this connection between GitHub and Zenodo that whenever you release the metadata, it's also persisted in Zenodo. So you get both of the sort of usability of a GitHub or similar solution and the long-term storage of metadata. So lastly, will be a small presentation about the interactions with the EDAM ontology, which is a life science-specific ontology by Claire Rioualen. See if I can start this.
Hello, my name is Claire Rioualen and I work for the French Institute of Bioinformatics in Marseille, France. My main area of work is the development of the EDAM ontology as a standard for the annotation and interoperability of resources in the domains of life sciences and computational biology. In the context of genomic analysis, one can come across a wide variety of data and metadata files, which are sometimes formatted in non-conventional ways or not properly annotated. The adoption of common standards for metadata annotation is crucial for the FAIRification of genomic data production, analysis, and interpretation. In this context, ontologies are a powerful tool for the uniformization of annotations and data description. Ontologies consist of control vocabularies where each term can be assigned properties such as a definition, synonyms, permanent ideas, and hierarchical and semantic relationships. The EDAM ontology defines and formalizes concepts for the domain of life sciences and computational biology that can be used in metadata schemas. Its vocabulary is structured into 4 main classes. Topics define scientific domains and technologies. Operations describe data processing functions. Data types represent objects that are produced or processed through operations. And formats are the serialized version of pieces of data into computer files. Those concepts and terms are assigned unique permanent URLs, guaranteeing the long-term validity of the metadata. In the context of the FAIRification of genomic tracks, the EDAM ontology can be used to describe and annotate many track attributes. For example, the topic ChIP-seq can be used to describe the type of experiment performed. Peak calling is an operation that processes genomic sequences and produces sequence features called peaks. Those peaks can be encoded in the form of bed files. These concepts can also be applied to software tools and workflows, and the annotated objects can be loaded into a genome viewer, provided they come in appropriate formats. Using this same example, this figure illustrates the annotations that can be used for genomic files metadata, keeping track of their origin and the processing steps they have undergone before being visualized and interpreted with the use of a genome browser. Overall, ontologies are a powerful tool to uniformize a large variety of data objects and allow their traceability and reusability. Beyond metadata description, they can also enable the integration of this data with more information and the construction of knowledge graphs. In the context of rapid advances in artificial intelligence technologies, ontologies can unlock more powerful applications. Those applications are potentialized by sharing standards among large communities and across interoperable resources. The EDAM ontology is already used within a wide ecosystem of life science-related knowledge bases, organizations, software tools, or web schemas.
Thank you. Yes, so that is really all the slides I have. I wanted to thank, well, there's supposed to be a thank you slide here, but that got lost in the process somewhere. Thanks a lot to the other co-chairs, Anna Bernasconi, Nathan Sheffield, Adam Wright, and all the other contributors, including Claire, that provided some comments from EDAM. So if you are, are there any questions, either from the audience or from the online? So once more, I will need to apologize for the place, or then any. And please join our working groups or take contact with me if there is any thoughts and ideas we want to collaborate somehow. Thank you for your attention, and enjoy the rest of the conference.